Hello, I'm SA, your AI essayist. Please note that all content, audio and video in this presentation are AI generated. As AI can sometimes produce inaccuracies, I encourage you to verify any factual information presented. With that in mind, let's begin.
The late 19th and early 20th centuries presented a significant challenge to those seeking to understand the human body. Neurobiology, a field in its early stages, faced fundamental questions. How does the brain generate thought, feeling, and action? What are the physical bases of consciousness? The scientific approach, while beginning to accept cellular theory, lacked the tools to study the complex networks of neurons in the nervous system. New methods were needed to observe and understand the brain's microscopic details. Basti VN, among others, sought to answer these questions, working within the limits of existing technologies and theories. This essay will discuss the contributions of Basti VN, a key figure in neurobiology. His work, though sometimes less recognized than that of others, greatly influenced our understanding of the brain's structure and function. We will follow the development of his ideas, examining the problems he addressed, the solutions he created, and the lasting impact of his research. The discussion will cover several key areas. First, we will examine his early life, education, and the intellectual environment that shaped his scientific views. Second, we will analyze his important work on neuronal form, especially his development of a new staining technique that allowed detailed viewing of individual neurons. Third, we will explore his contributions to understanding synaptic transmission, the process by which neurons communicate. Fourth, we will examine his research on the functional organization of the cerebral cortex, a brain region responsible for higher cognitive processes. We will assess his role in the development of the neuron doctrine, a fundamental principle of neurobiology. We will discuss the impact of his work on subsequent generations of neuroscientists and the relevance of his discoveries to modern research. We will consider the legacy of Basti VN and his enduring impact on the field. Basti VN's journey began in a period of intense scientific ferment, a time of rapid advances in biology, chemistry, and physics. Born in a small town in the Austro-Hungarian Empire in 1860, Basti VN displayed an early aptitude for scientific inquiry. His father, a physician, instilled in him a deep appreciation for the natural world and a keen interest in the workings of the human body. This early exposure to medical practice fostered a sense of curiosity and a desire to understand the complexities of disease and health. His formal education commenced at the local gymnasium, where he excelled in mathematics, physics, and the natural sciences. The rigorous curriculum provided him with a solid foundation in the principles of scientific reasoning and experimentation. He then pursued medical studies at the University of Vienna, a renowned center of scientific learning at the time. The university boasted a distinguished faculty, including several prominent figures in physiology and anatomy. The intellectual atmosphere was vibrant, characterized by lively debates and a constant exchange of ideas. This environment fostered a spirit of inquiry and encouraged students to challenge existing paradigms. During his medical training, Basti VN became increasingly fascinated by the nervous system. The brain, with its enigmatic nature and complex functions, presented a compelling scientific puzzle. Existing methods for studying the brain, primarily gross anatomical dissection, provided limited insight into its microscopic structure. The prevailing view, influenced by the reticular theory, described the nervous system as a continuous network of interconnected fibers, which impeded progress in understanding the brain's fundamental units. Visualizing individual neurons and their intricate connections presented a significant challenge. Available techniques, such as staining with dyes, often produced a dense, opaque mass, concealing the details of neuronal structure. Basti VN's initial research centered on developing improved histological techniques. He understood that the key to unlocking the brain's secrets involved visualizing its microscopic architecture with greater clarity and precision. He experimented with various staining methods, seeking a technique that would selectively stain individual neurons, enabling detailed examination. This period involved intense experimentation and meticulous observation. The scientific community anticipated a new approach. His early work, conducted in the late 1880s, established the foundation for his later, more significant contributions. He studied the work of other researchers and sought inspiration from diverse fields, including chemistry and microscopy. The scientific landscape evolved rapidly, and he was determined to be at the forefront of this transformation. Basti VN was poised to make his mark on a field. Basti VN's initial work was influenced by the constraints of existing histological practices. The late 19th century witnessed progress in microscopy, but studying the nervous system was difficult because individual nerve cells, or neurons, were hard to distinguish. Existing methods, which relied on bulk staining, created a tangled, unclear mass of tissue. The details of neuronal structure, including dendrites and axons, were hidden. Researchers found it difficult to understand the complex networks that formed the brain's structure. The main challenge was to develop a method that would stain neurons selectively, highlighting their individual forms and allowing detailed observation of their connections. This was a core problem that guided Basti VN's early research. Techniques of the time, such as carmine and hematoxylin, stained entire tissues, making individual cells unclear. The resulting images did not offer much clarity about neuronal shape or connections. The scientific community needed a method that would provide a clear and detailed view of the nervous system's microscopic structure. Existing methodologies were too broad failing to provide the necessary resolution to study the brain's complex organization. The inability to see individual neurons with precision slowed progress in understanding the fundamental principles of neural function and communication. Basti VN recognized the need for a more selective approach. Basti VN's unique solution came from his careful experimentation with various chemical reagents and staining procedures. He aimed to find a substance that would selectively attach to the cellular components of neurons, creating a high-contrast image under the microscope. He was dedicated to observation and understood the chemical properties of the materials he used. He carefully adjusted the concentration of stains, the duration of staining, and the methods of tissue preparation, exploring the parameters that influenced the staining process. His laboratory was a place of experimentation where he pursued selective neuronal staining. Basti VN's approach involved silver salts. He discovered that certain silver solutions, when applied to brain tissue, would stain a small subset of neurons, creating a contrast against the surrounding tissue. The mechanism of this staining process was, at the time, a mystery, but the results were undeniable. The silver salts, under specific conditions, would precipitate within the neurons, making them visible under the microscope. The method, known as the silver impregnation or silver staining method, provided selectivity previously unattainable. Basti VN's technique allowed researchers to visualize individual neurons, along with their intricate processes. The significance of Basti VN's staining method was its ability to reveal neuronal structure with clarity. The silver staining technique highlighted the dendritic trees, the branching extensions that received signals from other neurons, and the axons, the long, slender projections that transmit signals to other cells. The technique unveiled the architecture of the nervous system. This allowed for the study of neuronal morphology, the shapes and forms of individual neurons, and their connections. The ability to visualize these structures was a crucial step toward understanding how the brain functions. Basti VN's initial work profoundly influenced later research. His silver staining method offered a crucial tool, enabling detailed study of the nervous system's structure and organization. Other scientists adopted and improved the technique, establishing it as a standard method in neuroanatomical research for many years. The images generated using the silver staining method transformed scientific understanding of the brain. The method facilitated tracing nerve fiber pathways, identifying diverse neurotypes, and mapping the complex networks connecting brain regions. The silver staining method allowed for the study of the nervous system's cellular architecture. Researchers could classify neurons based on shape, size, and connections. For instance, the method enabled the identification of different neuron types, including pyramidal cells in the cerebral cortex. The ability to visualize these cells, with their unique shapes and elaborate dendritic trees, represented a significant advancement. The silver staining method provided the visual basis for studying brain structure and function. Basti VN's early research also significantly impacted the development of the neuron doctrine. This fundamental neuroscience concept proposes that the nervous system comprises individual cells or neurons, which communicate through specialized junctions later identified as synapses. The silver staining method provided the visual evidence supporting this doctrine. Images from Basti VN's technique demonstrated that neurons were individual cells separated by small gaps. This observation visually confirmed the concept of the neuron as the nervous system's fundamental unit. Basti VN's early work had a broad impact, extending into practical applications for studying neurological disorders. The silver staining method aided in identifying structural abnormalities in the brains of individuals with neurological conditions. This provided insights into the causes of these disorders, leading to new diagnostic and therapeutic approaches. The method became essential in studying diseases such as Alzheimer's and Parkinson's disease. Basti VN's meticulous approach, his dedication to visualizing the brain's microscopic structure, and his innovative use of silver salts contributed to his success. His work, occurring during advancements in microscopy and chemistry, advanced the field of neuroscience. The silver staining method, a result of his early research, provided the foundation for many subsequent discoveries. The method revealed the brain's intricate architecture, leading to a deeper understanding of its function. Basti VN's initial contributions were foundational to the evolution of neuroscience. His contributions to neuroanatomy, specifically his dedication to understanding the nervous system's structure, represent a significant advancement. He sought to understand the organization of neurons and their connections, a quest that required him to map the complex network of neural pathways, the communication lines that allowed the brain to function. Existing methods offered glimpses of the brain's structure, yet they did not provide a comprehensive picture of neuronal connections. Basti VN addressed the challenge of visualizing neural connections. He developed techniques to visualize these connections, a process now known as tracing neural circuits. The silver staining method, useful for individual cells, required refinement to reveal connections. He modified the staining process to highlight axons and dendrites, the extensions of neurons. This involved experimentation with chemical concentrations, staining times, and tissue preparation. The objective was to selectively stain neural fibers, making them visible. Basti VN's methods included innovative techniques. He introduced impregnation, allowing silver salts to bind to neural fibers. This was followed by a reduction process, causing silver to precipitate, forming a visible deposit. He also developed techniques for sectioning brain tissue into thin slices, enabling observation under a microscope. These sections were carefully prepared to minimize damage. Meticulous tissue preparation was essential for clear images. A key innovation was Basti VN's method to trace individual neuron pathways. By staining axons and dendrites, he could follow a single neuron's course through the brain. The method highlighted specific pathways, allowing him to trace their course. This approach enabled him to chart the relationships among various brain areas. The capacity to follow these connections was transformative, yielding a novel comprehension of how the brain handles information. Basti VN's contributions to neuroanatomy were substantial. His techniques enabled researchers to observe the intricate structure of the nervous system with unprecedented clarity. He created the initial detailed maps of neural circuits, unveiling the complex connections that govern brain function. This work directly influenced the study of the brain. Neuroscientists globally embraced his methods, facilitating the exploration of the nervous system in innovative ways. His work established the basis for contemporary neuroanatomy, which continues to utilize the principles and techniques he initiated. The methods Basti VN devised also significantly influenced related fields. His techniques were modified and improved for use in the study of neurodegenerative conditions, including Alzheimer's and Parkinson's disease. The capacity to visualize neural connections allowed researchers to investigate the alterations occurring in the brain during these diseases. The methods also offered a framework for understanding the mechanisms of neuronal damage and the creation of potential treatments. The understanding of neural pathways, initially developed through Basti VN's work, proved essential in the advancement of new diagnostic and therapeutic approaches. Moreover, Basti VN's work facilitated the development of new imaging techniques. His methods provided the foundation for the creation of more advanced imaging techniques, such as electron microscopy and confocal microscopy. The techniques, built upon silver staining principles, yielded enhanced resolution and detail. The methods he devised facilitated the development of novel imaging techniques. These new techniques have transformed the study of the nervous system. They have enabled researchers to examine the brain with unparalleled precision, offering fresh perspectives on its structure and function. Basti VN's dedication to understanding the brain's microscopic structure, along with his development of methods to visualize neuronal connections, proved crucial in advancing neuroscience. His work offered a new understanding of the brain's organization. His methods provided essential tools for studying the nervous system. His contributions have had a lasting impact on neuroanatomy and continue to influence research. His legacy is evident in ongoing efforts to map the human brain and to understand the complex workings of the nervous system. The techniques he developed, though refined over time, remain relevant in modern neuroscience. The central challenge that engaged Basti VN in the mid-20th century, and one that continues to resonate with researchers, was the need for a clearer, more precise understanding of how individual neurons connect. Existing methods for visualizing the brain's structure, while informative, were limited. These methods, often reliant on staining techniques, provided a general overview of the brain's architecture. They lacked the resolution needed to discern the intricate details of neural connections, the synapses, the minute gaps across which signals are transmitted. The brain, with its billions of neurons and trillions of synapses, presented an almost unfathomable complexity. Existing methods presented challenges in fully understanding the brain's intricate organization, yielding an unclear view of its functional aspects. Early techniques had several drawbacks. Staining methods, though helpful, frequently introduced artifacts, altering the original structures. The resolution was inadequate for differentiating individual synapses, preventing researchers from mapping neuronal communication pathways. Moreover, these methods were generally destructive, necessitating the preparation of non-living tissue samples. This prevented the study of the brain's dynamic activity in its living state. Precise tools were absent, hindering the correlation of structure and function, a vital step in understanding brain information processing. The situation resembled attempting to comprehend a large city using a poorly detailed map, unable to discern individual buildings, streets, or traffic flow. Basti VN aimed to address these limitations. He employed a novel strategy, utilizing the developing capabilities of electron microscopy. This technology, employing an electron beam instead of light to illuminate the sample, provided significantly enhanced resolution. The electron microscope could reveal structures at the nanometer scale, a previously unattainable level. This offered the potential to visualize individual synapses and trace neuronal connections. Basti VN's solution centered on a careful preparation of brain tissue for electron microscopy. The process demanded a thorough comprehension of chemistry, physics, and biology. The objective was to maintain the intricate brain structures, ensuring they could endure the powerful electron beam without altering the cellular elements. Basti VN devised a multi-stage procedure encompassing fixation, embedding, sectioning, and staining. The fixation stage, utilizing a chemical fixative, effectively stabilized the brain tissue, halting degradation and preserving its structure. Embedding involved encapsulating the tissue within a rigid, supportive substance, facilitating the creation of exceedingly thin slices. These thin slices underwent staining with heavy metals, such as osmium tetroxide, to scatter the electrons and generate image contrast. The innovation resided in the specifics of each stage. Basti VN meticulously refined the fixative, the embedding medium, and the staining protocols. He explored various chemical concentrations, exposure durations, and sectioning techniques. His aim was to attain the highest possible resolution while reducing imperfections. This meticulous attention to detail characterized his work. The careful preservation of the tissue was essential, requiring precision throughout the process, from the initial fixation to the final imaging. Basti VN's methodology was grounded in the fundamental principles of microscopy and the properties of matter. The electron microscope's capacity to reveal fine details depended on the wavelength of the electrons. Basti VN recognized that sample preparation was crucial for image quality. The fixative was essential to halt all activity, thereby preserving cellular structures in their original state. The embedding medium was necessary to support the tissue during sectioning. The staining process needed to introduce contrast without causing structural damage. This required a thorough comprehension of the interactions between electrons and matter. Basti VN's work had a profound impact, establishing the basis for contemporary neuroanatomy. His methods allowed researchers to visualize the intricate details of neuronal connections, revealing the structure of synapses for the first time. This facilitated a wave of new discoveries. Scientists gained the ability to study the brain at the level of individual neurons, mapping their connections and gaining insights into their communication. The correlation of structure and function became achievable. The techniques he developed found application in diverse studies, encompassing brain development and neurological disorders. His work directly contributed to the understanding of how the brain processes information. The identification of different synapse types, along with the analysis of their distribution, provided insights into the mechanisms of synaptic transmission. This information has been crucial in understanding how the brain learns, remembers, and makes decisions. His methodology also influenced materials science and engineering, fostering new methods for preparing and imaging samples. The significance of Basti VN's work persists. His methods continue to be employed in neuroscience research, evolving over time. Modern electron microscopy techniques, such as cryo-electron microscopy, are based on his foundational work. These techniques provide higher resolution, enabling researchers to study the brain in new ways, including studying the brain in its hydrated state. The effort to understand the brain's structure and function continues, and Basti VN's contributions remain essential. His work has provided the tools and the inspiration for generations of scientists to explore the mysteries of the most complex organ in the human body. The detailed mapping of neuronal connections, the study of synaptic plasticity, and the development of new treatments for neurological disorders all benefit from his innovative approach. Building upon advancements in sample preparation and imaging, Basti VN's contributions included a specific methodology that revolutionized the study of neuronal structures. This methodology, centered on the meticulous application of electron microscopy, addressed the limitations of earlier techniques. The challenge was preserving delicate neuronal structures during sample preparation, a critical step before imaging. Earlier methods often introduced artifacts, distortions, or destruction of the features researchers sought to observe. This hindered the ability to accurately visualize and interpret the intricate architecture of the brain. Basti VN's solution involved a multifaceted approach that improved the fidelity of electron microscopy. The core innovation involved a refined process of fixation, embedding, and sectioning. Basti VN advanced fixation techniques, employing a combination of fixatives and optimized protocols to improve results. He used aldehyde-based fixatives, including glutaraldehyde and formaldehyde, carefully managing their concentration and exposure time. This careful control helped reduce artifacts, preserving cellular components, original dimensions, and relationships. He then embedded the fixed tissue in a solid matrix, a necessary step for sectioning the sample into ultra-thin slices for electron microscopy. Basti VN improved the embedding process by using epoxy resins, which provided better structural support than earlier embedding media. Epoxy resins enabled the creation of extremely thin sections, often just tens of nanometers thick. These thin sections were essential for achieving the high resolution needed to see the intricate details of neuronal structures. The thinner the section, the clearer the image. The final stage involved an ultramicrotome, a specialized instrument for cutting these ultra-thin sections. Basti VN demonstrated a strong understanding of sectioning mechanics. He optimized the cutting knife's angle, the sectioning speed, and the environmental conditions. These adjustments significantly improved the section's quality, resulting in minimal compression, distortion, or damage. Basti VN's methodology was based on a deep understanding of the physics and chemistry of each step. He understood that the interaction of electrons with the sample was essential for image formation. He meticulously adjusted the electron microscope's settings, including the accelerating voltage and the beam current, to achieve optimal image contrast and resolution. He also understood the significance of staining the sections with heavy metals, such as uranium and lead, to increase electron scattering. These heavy metals selectively attached to cellular structures, enhancing contrast and making them visible under the electron beam. Basti VN's methods had a transformative effect. His improved protocols resulted in a significant enhancement in the quality of electron micrographs. Scientists could, for the first time, see the detailed structure of neurons, synapses, and other neural components with exceptional clarity. This improved visualization enabled the precise mapping of neuronal connections, a crucial step in understanding brain function. The methodology supported groundbreaking investigations into synaptic structure and function. Synapses, the junctions between neurons where signals are transmitted, are incredibly complex structures. Basti VN's techniques allowed researchers to study the morphology of synapses, measure the size of synaptic vesicles, and analyzed the organization of the presynaptic and postsynaptic densities. These microscopic observations provided valuable insights into neurotransmission and synaptic plasticity, the ability of synapses to strengthen or weaken over time. Moreover, his methodology aided the study of various neurological disorders by providing a way to examine the ultrastructure of the brain in disease states. Researchers could identify structural abnormalities associated with conditions such as Alzheimer's disease, Parkinson's disease, and stroke. The detailed visualization of cellular damage, the accumulation of abnormal proteins, and the disruption of neuronal connections provided critical clues to the underlying mechanisms of these devastating illnesses. Basti VN's work significantly impacted fields beyond neuroscience. Researchers in cell biology, developmental biology, and materials science adopted his methods. His pioneering work in sample preparation, fixation, and imaging proved useful in studying many biological and non-biological materials. Advances in electron microscopy techniques spurred progress in other imaging methods, including X-ray microscopy and atomic force microscopy. Basti VN's methodology was based on a thorough understanding of the physical and chemical properties of biological materials and the principles of electron microscopy. He approached the problem systematically, carefully considering each step and optimizing parameters for optimal results. His work demonstrates the importance of meticulous attention to detail, rigorous experimentation, and a deep theoretical understanding in scientific research. The methodologies he developed are still fundamental to modern neuroscience and continue to guide research into the brain's complex functions. The groundbreaking nature of Basti VN's work, with its novel techniques and ambitious scope, naturally attracted attention. Examining the microscopic architecture of biological structures, especially the brain, presented inherent challenges. The technology's limitations, the specimen's delicate nature, and the system's intricate complexity provided opportunities for discussion. Initial debates focused on image interpretation. The three-dimensional reconstruction techniques, though revolutionary, relied on assumptions and processing algorithms. Critics questioned the potential for artifacts during preparation and imaging. They suggested that the early techniques might be prone to distortions, possibly leading to misinterpretations of the underlying biological reality. The visual representations could be a product of the process as much as a faithful depiction of the structure. The debate concerned the accuracy of the images in reflecting the true form of the biological structures and the degree to which the methods might introduce artificial features. Basti VN and his team responded to these criticisms with meticulous detail. They conducted a series of rigorous validation experiments, comparing their results with other established methods when possible. They performed extensive controls, varying the preparation protocols and imaging parameters to assess the robustness of their findings. They also collaborated with other researchers, sharing their data and techniques to allow for independent verification. This collaborative approach, open and transparent, allowed for a broader consensus regarding the validity of the methods. They emphasized the importance of careful interpretation, cautioning against overinterpretation of individual images and advocating for the use of multiple lines of evidence. The team consistently stressed that the images were best understood within the context of the broader biological understanding. A second, more technical area of debate concerned the specific methods used to prepare and stain the specimens. The techniques for preserving and enhancing contrast in biological samples were, at the time, still evolving. The team faced the challenge of balancing the need to preserve the delicate structures of the brain with the need to generate sufficient contrast for visualization with electron microscopy. The use of heavy metal stains, crucial for enhancing contrast, presented the risk of introducing artifacts or damaging the delicate structures. The precise protocols for fixation, dehydration, and embedding were therefore subjects of intense discussion within the scientific community. Basti VN's team addressed these concerns by continually refining their protocols. The team investigated various fixatives, embedding media and staining techniques, meticulously assessing the effect of each on the final image quality. They documented their methods in detail, explaining the reasoning behind each selection, which enabled other researchers to reproduce and assess their work. They also worked to enhance the resolution and clarity of the images, aiming to reduce any potential distortions from the preparation process. The team's dedication to openness and their persistent pursuit of technical excellence were essential in establishing the validity of their approach. The team's research questions were ambitious, which also led to discussion. They aimed to understand the brain's functional organization in addition to its structure. This required them to connect structural features with physiological processes, a task of great complexity. The interpretations of the team's findings, particularly those concerning synaptic connections in neuronal circuits, were sometimes questioned. Some highlighted the limitations of drawing functional conclusions from static images, stressing the need for further experimental evidence to support their claims. Basti VN and his collaborators acknowledged the difficulties in interpreting static images. They emphasized that their structural studies provided a basis for understanding the brain's functional organization. They then integrated their structural findings with data from other sources, including electrophysiological recordings and behavioral experiments. The team also created new methods for connecting structural details with functional activity, such as combining electron microscopy with techniques for tracing neural pathways. The team's approach became a model for integrating multiple sources of evidence, showing the importance of combining structural and functional data to achieve a complete understanding of the brain. The discussions surrounding Basti VN's work ultimately strengthened it. The team refined their methods, validated their findings, and articulated their interpretations with greater clarity. Openness to criticism and engagement in rigorous scientific debate cultivated a culture of intellectual honesty and continuous improvement. These controversies served as a catalyst, accelerating the field's development. The impact of these debates extended to establishing rigorous standards for electron microscopy and three-dimensional reconstruction techniques. Validation, reproducibility, and open data sharing became hallmarks of the field. The debates also highlighted the importance of interdisciplinary collaboration, encouraging researchers from different fields to work together on complex scientific questions. The legacy of these debates is a stronger, more robust scientific discipline, valuing critical thinking, meticulous experimentation, and a commitment to truth. The controversies became a testament to the enduring power of scientific inquiry. The intellectual landscape, reshaped by Basti VN's endeavors, presents a testament to the power of persistent inquiry and the profound influence of dedicated exploration. Their contributions are multifaceted, extending far beyond individual discoveries. The profound shifts in perspective resulting from their methodologies and the impact on scientific thought were significant. Their work facilitated a remarkable transformation in the field. Challenges, complexities, and limitations of existing techniques were addressed with novel solutions, leading to the development of more sophisticated instrumentation and analytical methods. This propelled a cycle of innovation, where each advancement spurred further exploration and refinement. Rigorous standards for data acquisition and analysis guarantee the reliability and reproducibility of findings. These efforts continue to have an impact. Basti VN's legacy includes establishing a framework for scientific investigation. Precision, meticulous attention to detail, and a commitment to verifiable results became essential to the scientific process. This meticulous approach, combined with a willingness to embrace critical evaluation, fostered a culture of intellectual rigor. Their work encouraged a constant reassessment of assumptions and the pursuit of deeper understanding. Their influence extends beyond their immediate research domain. The methodologies they developed, the principles they championed, and the standards they established have found applications in diverse fields, including material science and biomedical engineering. The techniques and insights have been adapted and modified to address new challenges, demonstrating the remarkable versatility and enduring relevance of their contributions. The ripple effects, adaptations, and extensions of their work demonstrate the foundational nature of their discoveries. Basti VN's work catalyzed collaboration. Their research, involving complex instrumentation and sophisticated analytical techniques, required integrating knowledge and expertise from multiple disciplines. This collaborative spirit fostered a vibrant intellectual environment where researchers from different backgrounds could exchange ideas, share insights, and work together to address complex scientific questions. This synergistic approach accelerated discovery and spurred innovation. The long-term consequences of their work were evident in the evolution of scientific practice. Open data sharing, a commitment to reproducibility, and rigorous standards for data analysis are direct results of their efforts. These principles have helped protect the integrity of scientific research, ensuring findings can be validated and built upon by other researchers. Transparency and accountability have helped build public trust in science. Basti VN's work is undeniably relevant to contemporary scientific inquiry. The challenges of the modern era, including the need to understand and address complex problems, require the skills and methodologies that were pioneered. The ability to analyze vast data sets, develop sophisticated models, and integrate information from multiple sources is critical to advancing scientific knowledge. These are the tools and principles that were honed and refined in their groundbreaking work. Basti VN's place in the history of science is secure. They stand as a figure whose contributions have profoundly shaped the landscape of scientific inquiry. Their work embodies intellectual curiosity, the power of meticulous investigation, and the importance of rigorous standards. The legacy of their work extends beyond the specific discoveries, touching the very fabric of how science is conducted and how knowledge is advanced. Basti VN's contributions represent a triumph of the human intellect. They navigated the complexities of their chosen field with unwavering dedication, a relentless pursuit of truth, and a profound commitment to advancing scientific knowledge. Their work serves as an inspiration to future generations of scientists, reminding us of the power of human ingenuity and the enduring value of scientific inquiry. The impact of their work, the enduring relevance of their methodologies, and the profound influence on the scientific community are a testament to their remarkable achievements. Their legacy encompasses intellectual rigor, persistent innovation, and a deep commitment to scientific truth.
We really appreciate you tuning into this AI-powered presentation. Our goal is to push the boundaries of AI in content creation, and your input is invaluable. If there's a particular subject you'd like to see us tackle next, or if you have suggestions on how we could fine-tune our AI's creative prompts, please don't hesitate to leave your ideas in the comments section. We're excited to bring you more unique content very soon.